Hello all, myself Archana Rathi and I welcome you to everybody's favorite Physics Wallah. Today we are going to revise the chapter Principles of Inheritance. And again I'm saying no tensions, no worries, just listen carefully and you'll be ready for the term 1. So without delaying, let's start the lecture. According to the CBSE syllabus, the topics we are going to cover today are Mendelian Inheritance, Incomplete dominance, codominance, multiple allelism, inheritance of blood groups, Pleiotropy, polygenic inheritance, chromosomal theory of inheritance, sex determination, linkage, crossing over, Mendelian disorders and non-Mendelian disorders. So let's continue. Before starting the lecture, I would be discussing some important terms with you which would be using in this chapter, and the terms are, first of all, let's discuss the concept of dominance and the concept of recessiveness. What is a dominant recessive allele? To understand this, let's go to the original allele present in the population. Original allele present in the population. During the course of evolution, this original allele can mutate and forms another form of the gene, that is the recessive allele. So you can say recessive allele is the mutated form of the original allele. The dominant allele, which is the original allele, forms the protein and is responsible for the expression, but the recessive allele does not form the protein. So what we understand here, that a single gene can have two forms. A single gene can have two forms. One form is called as the dominant form and the another form is the recessive form. And the recessive form is generally the mutated form of the original allele. The mutated form of the original allele. Now, here we'll understand a new term, beta, and that is allele. Allele or allelomorphic pair. Allele or allelomorphic pair. So what you can say, that alleles are two different forms of a same gene. Two different forms of a same gene. Okay? That means if we take the example of plant height, suppose I am taking the example of the gene plant height. The plant height gene, what we write, the dominant allele, what we write it as? Capital T, and the recessive allele as small t. So, capital T is the dominant allele and small t is the dwarf allele of the same gene, So, these are the alleles of the same gene, dominant and recessive. Okay? Now, let's understand the concept of homologous chromosomes. Homologous chromosome, right? In a diploid organisms like us, the chromosomes occur in pair and the pairs are called as the homologous chromosome. And why we call them as homologous chromosomes? Because these chromosomes are similar in size, shape, and the kind of genes they have. Like if this chromosome has a gene plant height at this particular location, then this chromosome will be having the gene of plant height at this particular location only. Okay, so these are similar in size, shape, and the kind of genes they have. Okay, Now, let us understand the concept of homozygosity and heterozygosity. Homozygous condition and heterozygous condition. In the homozygous condition, the two forms of the same gene are similar. Understand, beta, that in a diploid organism, a single gene occur in pairs. That means a single gene have two copies in a diploid organism present on the homologous chromosome. And when these two copies are similar, then that condition is the homozygous condition. And when these two copies are different, then the condition is heterozygous condition. So capital T, small t, and capital T, small t. This is the heterozygous condition, and these two are the homozygous conditions. Now let's understand what is phenotype and what is genotype. Phenotype is the morphological appearance. Let's say I can see from outside and tell that the plant is tall or dwarf. So the external appearance or the morphological appearance is the phenotype of any organism. Morphological appearance. And genotype represents the genetic constitution of the organism, which we cannot see from outside. So it represents the genetic constitution. That means being a capital T, this is the genotype of an organism, which represents a homozygous dominant individual. Capital T, small t, this is the heterozygous dominant, and small t, this is the recessive individual. Okay, so phenotype and genotype. Now let's understand the difference between character and trait. Suppose, for example, I am taking the example of plant height. Plant height is a character, and the two forms in which it can exist represents the trait. That means tallness, being tall, and dwarfness, that is being dwarf, are the traits, and this is the character. Likewise, I can take the example of flower color. Violet flower, being violet in the condition is a trait, and white is also a trait. So this is the difference between a character and a trait. And yes, one more term which is important, pure line or true breeding variety. Pure line or true breeding variety. Understand this, any individual which is homozygous for a trait is a pure line. Suppose this capital T is a pure tall plant, homozygous individual. So this is the pure line. Likewise, small t, it's a dwarf individual. It's also a pure line. That means homozygosity represents the pure line or the true breeding variety. Okay. So until now, what we have discussed, what is a dominant allele, what is a recessive allele, what is allele, different forms of the same gene. And in a diploid individual, we have two copies of the same gene. What is heterozygous, what is homozygous, what is genotype, what is phenotype, what is a character, what is a trait, and what are the pure lines. Now, let's go further. Now, let's discuss the first topic, that is Mendelian inheritance. Gregor Johann Mendel is called as the father of genetics, and he worked on Pisum sativum, as we all know, and he worked for 7 years. This could be a multiple choice question. He worked for how many years? For 7 years. from 1856 to 1863. Remember this, okay? And why his work was unique? Because he was the first one to apply the mathematical and the statistical analysis in the field of biology, and his work, observation and experiments were not based on a short sample size, but it was based on sample size. And one more thing, Pisum sativum generally, naturally shows self-pollination. You all know what is self-pollination. But in his experiments, Mendel did artificial hybridization also. And how he did artificial hybridization? By the process of emasculation. What is emasculation? Removal of anther from a bisexual flower before maturation. And that is how he did the cross-pollination. Now, Pisum sativum, garden pea, it is also a diploid plant like us, and it has 7 pairs of chromosomes, that is 14 chromosomes. And how many true breeding varieties Mendel selected for his experiment? 14. Remember this, 14 true breeding varieties. And now you know what is a true breeding variety or a pure line? Homozygous individual, whether dominant or recessive. Now, we have studied this in 10th class also. What are the reasons why Pisum sativum was taken? Because lifespan was short, it is easy to grow in the lab conditions. But the most important reasons are that there is presence of contrasting traits. Naturally, in Pisum sativum, there are contrasting traits, like some of the varieties are tall, some of the varieties are dwarf, some of the varieties have violet flowers, some of the varieties have white flowers. So presence of contrasting traits is the most important point. And generally, the Pisum sativum shows self-pollination, and because of the continued self-pollination, what we get pure lines or true breeding variety. We get the homozygous individuals or the true breeding varieties because of the self-pollination shown by Pisum sativum. And there's a line in NCERT that these pure lines or true breeding varieties, they show stable inheritance. And we have a choice of cross-pollination also by artificial hybridization. Now, this is a chart given in NCERT beta. And what is important to learn in this chart, that which trait is dominant and which trait is recessive. This is important. In case of seed shape, round shape is dominant, wrinkled is recessive. Learn with me. Seed color, yellow is dominant, green is recessive. In the case of flower color, violet is dominant, white is recessive. Pod shape, inflated or being full. Inflated condition is dominant and constricted condition is recessive. Pod color, green is dominant, yellow is recessive. Flower color, axial. Axial means sidewise, laterally, and when the flower is present on the tip, that is on the terminal end, that is the terminal flower position, which is recessive. And stem height, tall is a dominant and dwarf is recessive. Now, where there is a scope of confusion? In the color. Remember, in the seed color, yellow is dominant, and in the pod color, green is dominant. Do not get confused. Pod color, green dominant, seed color, yellow dominant. Now, there is an important thing which needs to be remembered here, that is recessive trait can express itself only in homozygous condition. This line is important. Understand this. A recessive trait can express itself only in the homozygous condition, because a recessive allele, when present with the dominant allele, it cannot give the expression because the dominant allele dominates it. So, and what we can say otherwise, that a dominant trait can express itself both in homozygous and heterozygous condition. Both in homozygous and heterozygous conditions. What does it mean? What does it mean? If I say that white flower color, the trait white, can express itself both in hetero and homozygous condition, that statement would be false, because white color is a recessive trait and it can only express itself in homozygous condition. But all the dominant traits here, they can express themselves both in heterozygous and homozygous. Okay? Because dominant allele can express itself both in homozygous condition as well as heterozygous. So these kind of question can come from this part. So remember this point. Now, let's discuss the experiments done by Mendel. First of all, the monohybrid cross, or one gene in. These are the crosses in which we study the inheritance pattern of only one gene. So the basic monohybrid crosses, capital T crosses with small t, small t. So what needs to be remembered here, Vita, that the parents are always the pure lines or the true breeding varieties. These are the pure lines or the true breeding varieties. And the F1 generation, the hybrid is the hybrid. Now, if I say that F1 is phenotypically and genotypically similar to one of the parents, this would be false or true? Think. This would be false, because the F1 hybrid is genotypically dissimilar from both the parents, but it is phenotypically similar to one of the parents, and that is the dominant parents. And there is no ratio in the F1 generation, because in the F1 generation all are tall, heterozygous tall, all are heterozygous tall. And then what we do? We do selfing. And in the selfing, we cross the F1 hybrids together. That is, their genotypes and phenotypes should be same. This is called as selfing. The type of gametes they can form is small t, capital T, small t. Now Mendel, what was unique about the Mendel's experiments? That he used Punnett square to deduce his experiments. Now we will put one type of gametes here and the other type of gametes here. So capital T, small t, and small t, small t. So these are the basic things you already know. Okay, so this, what we have obtained here, beta, it's a F2 generation. Now what you can see here, beta, in the F2 generation, that we have two types of phenotypes in the F2 generation. In the F1 generation, there was only one phenotype, that is tallness. But in the F2 generation, there are two phenotypes, tall and dwarf. And how many genotypes? The genotypes are 3. And those genotypes are capital T, small t, and small t. So this is a direct question which comes, that in a monohybrid cross in F2 generation, how many genotypes and phenotypes are possible? Then 2 phenotypes and 3 genotypes. Now let us talk about the phenotypic ratio. phenotypic ratio in the F2 generation comes out to be 3:1. What is the phenotype? The outer appearance. So, this individual is tall, this is tall, this is tall. So, how many tall? 3. And how many dwarf out of 4? So, the ratio comes out to be 3:1. And the genotypic ratio comes out to be 1:2:1. Because how many genotypes? 3. Individuals, these type of individuals 1, these type of individuals 2, and these type of individuals 1. So the ratio 1:2:1. So these were the findings or conclusion of the monohybrid cross. Let's move on further. Let's discuss a back cross now. A back cross is a cross of F1 individuals with either of the parents. You can take any parent and cross it with F1, then it would be a back cross. And back cross is further of 2 types. That is test cross and out cross. A test cross is a cross of F1 with the recessive parent. And out cross is the cross of F1 with the dominant parent. The dominant parent. Okay. So if I say every out cross is a back cross, would that be false or true? Think it would be true. Every out cross is a back cross, but every back cross is not an out cross because it is of 2 types, test cross and out cross. So let's move on with the test cross. If I have to define a test cross, then the definition is the cross of F1 with the recessive parent is test cross. But the utility of test cross in the field of genetics is very wide. That means a cross of any individual with the recessive individual is a test cross. Mendel used the test cross in the F2 generation. He had tall individuals and dwarf individuals in the F2 generation. He wanted to know that these tall are capital T tall or capital T small t tall. So he used test cross for those questions. Now, test cross. Let's first describe a test cross. I am saying that it is a cross of F1 with the recessive parent. So, beta, what will be the condition? Capital T, small t, small t, small t. So, capital T, small t. This comes out to be small t, capital T, small t, and small t, small t. Right? Now, listen carefully. How many phenotypes you can see here? How many phenotypes? These will be tall, these will be tall, these will be dwarf, these will be dwarf. So how many phenotypes you are getting in a test cross? 2 phenotypes. And how many genotypes you are getting in a test cross? 2 genotypes. See, difference. In a monohybrid cross, there were two phenotypes and 3 genotypes. But here in the monohybrid test cross, this is a monohybrid test cross. You have 2 genotypes and 2 phenotypes. And the ratio, the phenotypic ratio, how many phenotypes, beta? 2. Tall or dwarf? How many tall? 2. How many dwarf? 2. So the ratio comes out to be 1:1. And the genotypic ratio, capital T, small t, 2. Small t, 2. So the ratio comes out to be 1. Remember this thing. This is very important, that in a test cross, genotypic ratio is equal to phenotypic ratio, and that is 1:1. 1:1. Now, what is the utility of the test cross? The utility of test cross is to check the heterozygosity or homozygosity of the plant with unknown genotype. To check heterozygosity and homozygosity of plant with unknown genotype. Of plant with unknown type. Like Mendel did in the F2 generation, there were tall plants, but he did not know which plant is capital T tall and which plant is capital T small t tall. So we want to know that, and to know that we can take the help of test cross. How? Let's see, if the genotype of the unknown plant, genotype of the unknown plant was capital T, and I'm crossing it with the recessive individual, then what will be the result, beta? What will be the result? All will be tall. All will be tall. And if suppose the genotype of the plant was capital T small t, then what will be the result? The result will be capital T small t, capital T small t. So you can see here, beta, that in this condition, half of them are tall and half of them are dwarf. That means, how will you get to know that the plant whose genotype we want to know, whether it is homozygous or heterozygous, how will you tell? You will tell by seeing the results. If after the cross of the plant with the unknown genotype with the recessive individual produces all dominant individuals, that means all of them were tall. That means the plant was capital T tall. And if the result were showing that half of the individuals were tall and half of the individuals were dwarf, and the plant's genotype was capital T small. So this is how test cross is used to understand, to know the homozygosity and heterozygosity. Now let's move further. And yes, one more thing, beta, that we can also understand here that what is a dihybrid test cross. The dihybrid test cross. A dihybrid test cross. See, understand one thing. A dihybrid cross can be a multiplication of two monohybrid crosses. Okay, so a dihybrid test cross would be a multiplication of two monohybrid test cross. Monohybrid test cross to monohybrid test cross. That means the ratio of one monohybrid test cross is 1:1 and the another one is 1:1. So it comes out to be 1:1. Understand this. So in a dihybrid test cross ratio, the PR is equal to GR, and that comes out to be 1:1:1:1. And in a dihybrid test cross ratio, the number of genotypes would be 4 and the number of phenotypes would also be 4. Okay, so in a test cross, number of phenotypes, number of genotypes are same and ratios are also same. Now let's move on to the dihybrid cross or the two gene inheritance. In the monohybrid cross, we were seeing the inheritance pattern of only one gene, but here the inheritance pattern of two genes are studied together. So here we are taking two genes together, right? So this is round yellow crossing with the wrinkled green. So these are also the pure lines, as in Mendelian cross. This is also a homozygous individual. This is also a homozygous individual. And the F1 would be this. This is the F1, the hybrid. The hybrid. And here you can see the hybrid is heterozygous for both the traits. Hybrid is heterozygous for both the traits. And then selfing again. And after the selfing, how many zygotic combinations we will be getting in the F2 generation? We will be getting 16 zygotic combinations or offsprings in the F2 generation. And these are these. You can see the Punnett square here, beta. Okay, now to simplify this, what we can do, just now I told you that a dihybrid cross can be a multiplication of two monohybrid crosses. Let's use that. Just write one character first. Okay. This is one trait. And I have written all the three forms of this trait. And I know the ratio also. What was the ratio, beta? 1:2:1. Okay. Now write all the three forms of the other trait in front of this. Capital Y, capital Y, capital Y, small y, small y, small y. Likewise here, capital Y, capital Y, capital Y, small y, and small y, small y. Here also, capital Y, capital Y, capital small y, and small y, small y. Okay, and the ratio we know, 1:2:1. 1:2:1. 1:2:1. Okay, now what you have to do, just write capital R, capital R, capital R first. Capital R, capital R, capital R, capital R, capital R, capital R. Right? Then just write capital R, small r, capital R. Then write small r, small r, small r. Okay? And then write the condition of Y, capital Y, capital Y, capital Y, small y, small. Again here. Okay. Now multiply the numbers, beta. Multiply the numbers. 1 ones are 1. 1, 2, so 2. 1s are 1. तो क्या आ गया? 1. तो 1:2:1. And 2 ones are 2. 2, 2, 4. 2 ones are 2. 1 into 1, 1. 1 into 2, 2. 1 into 1, 1. Right? So when you even close your eyes, you will see that in the uppermost three conditions, there was capital R, capital R, capital R. In the middle three conditions, capital R, small r, capital R, small r, capital R, small r. And in the below three conditions, small r, small r, small r. Right? And with them, you just have to write capital Y, capital Y, capital Y, small y, small y, small y. And in the above three conditions, the ratio was 1:2:1. In the below three condition, 1:2:1. And in the middle, 2:4:2. Okay. So what are these? These are the genotypes, and the genotypic ratio hence comes out to be 1:2:1 2:4 2 to 1:2:1. Right? So what you can see here, that these individuals would be round, 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 and these would be wrinkled, wrinkled, wrinkled. Yellow, yellow, these green, yellow, yellow, green, yellow, yellow, green. Okay, so how many phenotypes and genotypes you get from here, beta? The number of genotypes here, you can see the number of genotypes are total 1, 2, 3, 4, 5, 6, 7, 8, 9. Genotypes are 9. And how many phenotypes? How many phenotypes? phenotypes are total 4. Which ones? Round yellow, round green, wrinkled yellow, and wrinkled green. Okay, G. So 9 genotypes and 4 phenotypes. Again, a dihybrid cross is a multiplication of 2 monohybrid cross. In the monohybrid cross, the number of genotypes were 3. Here, 9. 3 into 3, 9. In the monohybrid cross, number of phenotypes were 2. Here, 4. 2 into 2, 4. Okay, remember this. The genotypic ratio comes out to be, you do not have to cram this ratio. If you will multiply 1:2:1 into 1:2:1, what will come? This will come. Okay. Likewise, the phenotypic ratio of a dihybrid cross can be obtained like this: 3:1, 3:1. That comes out to be 9:3:3:1. Okay. So the genotypic ratio is 1:2:1:2:4:2:1:2:1, and the phenotypic ratio is 9:3:3:1. So this is a dihybrid cross. Now let's move on to laws of inheritance. So based on the crosses he proposed laws of inheritance. Law of dominance and law of segregation are based on monohybrid cross. Remember this, beta. And the law of independent assortment is based on a dihybrid cross. Okay. So first, let's understand the law of dominance. And we know that in an individual, in a diploid individual, one gene has 2 copies, and in a heterozygous individual, one is dominant, one is recessive. So let's see what are the lines of NCERT, because these lines of NCERT can become the MCQ. So it says characters are controlled by discrete units called factors. Mendel did not know what are genes, chromosomes, and DNA. He just used the term factors, and what we call them today, gene. Okay. So these are a separate independent units called as factors, which controls a character, which are responsible for the expression of the character. And these factors in a diploid individual, they occur in pairs. I just told you that in a diploid individuals, a gene has 2 copies, a factor has 2 copies. That is why it occurs in pairs. And in a dissimilar pair, what it is saying, in a dissimilar pair, one of the copy always dominates the other. So one which dominates is a dominant allele, and one which is always being dominated is a recessive allele. Okay. And this says that the law of dominance is used to explain the expression of only one of the parental characters in the F1 hybrid. In the F1 hybrid, which was heterozygous, remember, it shows only the phenotype of the dominant parent, because the dominant allele, even if it is present in the single copy along with the recessive allele, it can produce the expression. And the ratio we obtain, that is 3:1, is also because of dominance. Okay, so these 3 lines can directly come, and they can the question can ask this line belongs to which law. So the ratio 3:1 obtained in the F2 generation can be explained with the law of dominance. Now let's move on to law of segregation. Segregation means separation. Now we understand this, that in the 11th class we have read the process of meiosis. In a diploid individual, during gamete formation and meiosis, there used to be separation of homologous chromosomes during anaphase 1, and then separation of chromatids during anaphase 2. So in a diploid individual, during gamete formation, there is separation of chromosomes. of homologous chromosomes, and along with it, the genes present on the chromosomes also gets segregated. The 2 copy of the same gene present on the homologous chromosome, they also get segregated during gamete formation, such that each gamete receive only 1 copy of the gene. That means one gamete can have either capital T or small t, never together. So that is why this law of segregation sometimes is also called as the purity of gametes. The law which explains purity of gametes. That means a gamete is pure for an allele. For an allele, there would only be 1 copy of the gene present in the allele. So this is law of segregation. Let's see what NCERT is saying. This law is based on the fact that the alleles do not show any blending. Alleles do not show any blending, and the characters are recovered as such in the F2 generation. That means in the F1 generation, that is capital T, small t, there is no blending. If the parent, the capital T parent, if suppose it was 10 centimeter long, then the this F1 hybrid in the F1 generation will also be 10 centimeter long. There will be no blending. There would be no intermediate condition that it will be 5 centimeter long or 6. No, it will be 10 centimeter long. So there is no blending. There is no mixing, and that is why these characters are recovered as such in the F2 generation, and that is why we get 2 types of phenotypes in the F2 generation, tall as well as the dwarf. Okay, so this is the law of segregation. Now, the law of independent assortment. This is based on the dihybrid cross. What does it say? It says that, let's read the lines of NCERT first. It says that when 2 pairs of traits are combined in a hybrid, understand, when 2 pairs of traits are combined in a hybrid, right? 2 pairs of traits are combined in a hybrid, then segregation of 1 pair of characters is independent of the other pair of characters. That means 1 pair will segregate independently of the other pair. They will not interfere or influence each other in the segregation pattern. Okay, now let's understand this. I'm going to say a line, just listen it carefully. Independent assortment holds good only when the 2 genes we are talking about are present on different non-homologous chromosomes, and it fails when these 2 genes are present on the same chromosome. Understand? Very important. When these are the homologous chromosomes having similar size and same kind of genes, beta. So if these are the other pair of, this is 1 pair of homologous chromosome and this is other pair of homologous chromosome, right? So I'm taking a hybrid. Okay, this is a hybrid having 2 pairs of chromosomes. The independent assortment is applicable only when these 2 genes are present on different non-homologous chromosomes. These are non-homologous chromosomes and these are homologous chromosomes. These are homologous chromosomes and these are non-homologous chromosomes. Okay, so what I'm saying that independent assortment can be seen only when they are present on different non-homologous chromosomes. If they are present on the same chromosome, yeah? If they are present on the same chromosome, then independent assortment fails. If they are present on the same chromosome, then independent assortment fails. Why? Why? Because of the phenomenon linkage. The genes present on the same chromosome, they show linkage. They always get inherited together, right? So they will not be freely segregating from each other. Okay. So the law of independent assortment, what we can write that law of independent assortment law of independent holds good when two genes are present on different non-homo chromosomes. Not be present. And where it fails when the two genes are present on the chromosome, right? Because here what we will see linkage, and due to linkage this independent assortment will fail. Okay, so let's move further. Now before talking the deviations from Mendelism, let's discuss some formulas. If I have to calculate the number of gametes formed by any individual, how will I calculate? If I have to calculate the number of gametes, the formula is 2 raised to the power n, where n is the number of heterozygous conditions. N is the number of heterozygous conditions. Now, how we will solve these type of questions? See, suppose there is an individual whose genotype is this. Okay. It is given in the question that this is an individual and you have to tell how many gametes are formed by this individual. So you will count the number of heterozygous conditions. Let's count. This is heterozygous condition, this is heterozygous condition, and this is heterozygous. So how many number of heterozygous conditions? 3. 1, 2, 3. So formula, let's apply the formula. 2 raised to the power 3 comes out to be. So this individual can form 8 gametes. So you can solve any problem of this kind. Now, suppose this is given that how many gametes would be formed by this individual. So how many heterozygous condition? 2. So 2 raised to the power 2 comes out to be 4. Okay. Then if you have to find out the number of zygotic combinations or offsprings possible, number of zygotic combination offsprings possible, how will you calculate? You will simply calculate it by gamete from parent 1, gamete from parent 1 into gametes from 2. Okay, G. How? Let's apply this formula with the help of an example. Suppose I'm taking 2 parents. The genotype of one of the parents is this, and the genotype of other parent is this. And we have to calculate that if these two are the parents, then what are the number of zygotic combinations of offsprings possible? So calculate the gamete from parent 1. You know how to calculate. Count the number of heterozygous condition. 2 raised to the power 1 comes out to be 2. Here also the number of heterozygous conditions, only 1. 2 raised to the power 1 comes out to be 2. So 2 into 2 comes out to be 4. So if these two are the parents, then the total number of zygotic combinations possible are 4. And then any example you can take. Suppose If these two are the parents, then calculate the number of zygotic combinations possible. So 1, 2. 2 heterozygous conditions. So 2 raised to the power 2, 4. Only 1 heterozygous condition. So 2 raised to the power 1 comes out to be 2. So 8. Okay. So this is how you calculate the number of zygotic combinations of offspring. Then let's discuss another type of questions. Come. Yeah. Suppose this is given. Is given that these are the two parents, and you have to calculate that what is the probability that this kind of offsprings will be generated by these parents. You have to tell what proportion of these individuals would be possible if these two are the parents. How we calculate? Yes. For this, we need to know the frequency method. We need to know the frequency method. What does it say, beta? If this condition is there, capital A is crossing with small a, right? Right? So all the individuals will be capital A, small a. So what will be the frequency? 4 by 4, that comes out to be 1. 4 by 4, that comes out to be 1. Then, if this is the condition, one is homozygous dominant, and the other is heterozygous, then obviously out of 4, 2 will be capital A, 2 by 4. You can see it by drawing the Punnett square also, and half will be this. So I can write it as 1 by 2 and 1 by 2. And again, if the condition is this, homozygous recessive crosses with heterozygous condition, then obviously half of them would be small a, and half of them would be capital A, small a. Okay, but if both of them are heterozygous, then obviously we will be having how many conditions? We will be having 3 conditions: capital A, capital A, small a, and small a, small a. And what would be the probabilities of these? Capital A, 1 by 4, capital A, small a, 2 by 4, and this is 1 by 4. Okay, beta. I will not simplify this 2 by 4 to 1 by 2. Why? Because here there are not 2 chances. There are 3 chances. Capital A, small a, and small a, small a. So remember this. Whenever both the conditions are heterozygous, then you will be having 3 chances: capital A, small a, and small a, small a. And the frequencies would be 1 by 4, 2 by 4, 1 by 4. Remember this thing. But if I have only 2 chances, that is small a, and capital A, small a, then 1 by 2 and 1 by 2. Here also 1 by 2, 1 by 2. If all are same, then 1. Now, let's clear this numerical by applying the frequency method. Right. Clear है? Yes. So beta, we have to solve this problem now. Okay. So imagine in your head, I'm writing what you should imagine in your head. This is a trihybrid cross. Break this trihybrid cross into 3 monohybrid crosses rapidly in your mind. Capital A, small a crossing with small a, right? Capital B, small b crossing with small b, small c crossing with capital C, small c. This I have done in my mind. I have broken this whole cross into 3 monohybrid crosses, right? Right? Capital A, small a, crossing with small a, capital B, right? Now, see beta, one is heterozygous, one is homozygous. So, half of the probability would be of this and half of the probability would be of small a, small a. Now, one is heterozygous, again, one is recessive. So, half of the probability will be of capital B, small b, and half of the probability will be of small b, small b. Now, small c crossing with capital C, small c, again, half of the probability would be of small c, and half of the probability would be of capital C, small c. Okay. Now, what we have to find out the probability of these individuals. So, let's apply the formula. Small a, frequency 1 by 2. 2. Okay. Capital B, small b, frequency by 2. Small c, 1 by 2 comes out to be 1 by. So what is the probability of this genotype? 1 by 8 if the parents are these. And if you want to know that whether you are putting these frequency right or wrong, then you can check it how? That the denominator which is coming over here, beta, the denominator which is coming over here is by default the number of zygotic combinations you will get by crossing these two parents. Okay, let's see, are we right or are we wrong? So let's check. How many heterozygous condition? 2. So 2 raised to the power 2 comes out to be 4. How many heterozygous condition? Only 1. So 2 raised to the power 1 comes out to be 2. So multiplication gives us 8. Gives us 8. So if these are the two parents, then the total number of zygotic combination or offsprings which are possible can be 8. And see what is there? 8. That means we have not put any frequency wrong. Clear? Clear? This is frequency method, beta. And this is very simple. If these two conditions are there, then all of them would be capital A, small a, 1. If one is homozygous, one is heterozygous, then half of them would be homozygous, same, and half of them would be heterozygous. Again, here the same. But if both are heterozygous, then 3 situations and the frequencies would be 1 by 4, 2 by 4, 1 by 4. Let's solve another question. Suppose this is given, these are the two parents. Okay, these are the two parents, and the question is saying that you have to find out the probabilities of these genotypes. A, B, C, okay. Let's find out the probabilities of these 4 genotypes when these two are the parents. But let's do one thing. Let's calculate the number of zygotic combination rapidly. How many heterozygous condition? 1. So 2 raised to the power 1 comes out to be 2. How many heterozygous condition? Only 1. So 2 raised to the power 1 comes out to be 2. Okay. So how many total number of zygotic combination and offsprings are possible, beta? 4. So if we'll not put any frequency wrong, then in the denominator by default 4 will come. By default 4 will come. Let's do. So these are the two parents, and I have broken this dihybrid cross in my mind into 2 crosses. This will be very fast. Once you'll practice it at home, this will be very fast. Okay. So see, capital R, small r is crossing with cap, right? So capital R 1 by 4, capital R small r 2 by 4, and small r 1 by 4. See, what will be the frequency of capital R small r? 2 by 4. Small y is crossing with capital Y. All of them would be capital Y, right? All of them. So, is this condition possible? No. So, 0. Small r is possible? Yes. What frequency? 1 by 4. Is this possible? Yes. What frequency? 1. So comes out to be 1 by 4. We have not done any mistake because by default 4 is coming. Okay. Then capital R small r is frequency 2 by 4. Small y capital Y frequency 1 comes out to be 2 by 4. Capital R small r frequency 2 by 4, but capital Y is not possible, so 0. Okay, so applying this frequency method, you can solve any question of this sort, and we'll be doing more number of questions on this topic, the MCQ. So let's move forward. Now beta, let's discuss now the deviations from Mendelism. The law of dominance used to say that in a diploid individual, in a dissimilar pair of factor, one is dominant, one is recessive. But there is something which is not following this, and this is incomplete dominance and codominance. Incomplete dominance and codominance. But before that, I would like to take the topic of concept of dominance in detail. Okay, let's see. I am taking a original allele, which is usually called as the wild allele or the dominant allele. Okay, original allele, wild allele, or the dominant allele. Now, during the course of evolution, this original allele undergoes mutation. This original allele undergoes mutation. And what can be the results of this mutation in the original allele? There can be 3 conditions. First, if this mutated allele, even after mutation forms the same protein. Okay, what I'm saying, you will just understand the point. Understand. This is an original allele, and we also call it as dominant allele or the wild allele. This has undergo mutation, right? And we know that a dominant allele, a dominant wild or original allele, it forms the functional protein. That protein is responsible for the expression. Okay, so dominant allele forms the functional protein which is responsible for the expression. This dominant allele undergoes mutation, and even after mutation, this mutated allele forming the same functional protein and is producing the same phenotype, then this mutated allele is similar to the dominant allele. This is similar to the dominant allele. Okay, it will produce the same phenotype. But if the mutation has occurred and this mutated allele now no longer forms the protein, then this mutated allele is called as the recessive allele. Or it forms the protein, but that protein is of no use. That means non-functional protein. Then again, it is a recessive allele. Means if the mutated allele either do not form any protein or form the non-functional protein, then this mutated allele is the recessive allele. Okay, and this recessive allele is generally the mutated allele. Okay, third case, this mutated allele after mutation forms the protein, but the protein is altered, the protein is changed and is responsible for different expression, then that is a codominant. So how many conditions? 3. First, this mutated allele forms the same protein, then it will still be called as the dominant allele. Then it will still be called as dominant allele because there is a silent mutation here. What is a silent mutation? That even after the mutation, the codon codes for the same amino acid, because you know that many codons can code for the same amino acid. So even after the mutation, the mutated codon still coding for the same amino acid. So if there is silent mutation, then the mutated allele will remain dominant and will produce the same protein, and it will not affect the phenotype or the expression. But if this mutated allele either do not form a protein, or forms a non-functional protein, then in both the cases, the allele is said to be the recessive allele. And in the third case, if the mutated allele forms a protein, but that protein is altered, that protein is changed, it is not the similar protein as it used to form, but this is forms a protein. Yeah, you can say it forms an altered protein. It forms an altered protein. This explains the codominant. Okay? Codominant allele. Understand this. Understand this. Original allele, wild allele, after mutation, if the mutated allele still forms the same protein, then it is still called as the dominant allele. And if the mutated allele do not form a protein or form a non-functional protein, then this is a recessive allele. And if the mutated allele forms a changed functional protein, then it is a codominant. Okay. Now let's see what NCERT has to say about it. See, in the first case, the modified allele is equivalent to the unmodified allele. Got the line? The modified allele is equivalent to the unmodified allele, and it will produce the same phenotype or trait. That is, result in the transformation of substrate S. That means the protein will form its function, will continue its function, and will transform the substrate S into the product. Now see, such equivalent pairs are very common in a population. What kind of equivalent pairs? Where this original allele is the original allele, but mutation has occurred, but still this is called as the original allele because the mutation is silent and is producing the same expression. These kind of equivalent pairs are very commonly found in the population. But if the allele produces a non-functional enzyme or no enzyme, then the phenotype may be affected. Yes, it is affected. The tall plant becomes dwarf. The phenotype or trait will only be dependent on the functioning of the unmodified allele. Yani ke the dominant allele decides the expression which is the unmodified allele. But the modified allele, which is usually a recessive allele, will not produce the expression. Okay? The unmodified or the functioning allele, which represents the original phenotype or the original allele, is the dominant allele. As we have discussed, original, wild or dominant allele. And the modified allele, the changed allele, is generally the recessive. Okay? So let's move further. Yes. Now let's understand the deviation from the Mendel's law, that is incomplete dominance. The example of incomplete dominance can be seen in the inheritance of flower color in the plant Mirabilis jalapa, which is also called as the 4-o'clock plant, or it can be seen in Enterhinum majus, that is snapdragon or dog flower. Okay. Now, what is incomplete dominance? See, if we take the example of Mirabilis jalapa or snapdragon, these are the pure lines, the parents, right? Red, the flower color is red, and here the flower color is white. F1 hybrids are obtained, right? According to Mendel, what should have been the color? It should be red, but here what comes? Pink. What comes? Pink. So what is incomplete dominance? That in a paired condition where two alleles are dissimilar, the dominant allele is unable to show complete dominance. In the presence of recessive allele, the dominant allele is unable to show complete dominance, and hence it is incompletely dominating, and the color is not red but some of intermediate color, that is pink, and this is blending. Right? There was no blending in the Mendelian inheritance. Capital T, small t were also 10 centimeter long, and capital T were also 10 centimeter long. But here you are getting the intermediate condition, that is pink. That means this is not a completely dominant allele, and this is incomplete dominance. Now, again, selfing. And after doing selfing, what we used to obtain? We used to obtain how many genotypes in the F2 generation? 3. Capital R, capital R, capital R, small r, or small r, small r. Okay? But what's the difference here? That capital R, small r is red. Sorry, capital R is red, capital R, small pink, and small r is white. Understand. In the Mendelian monohybrid cross in the F2 generation, we used to have 2 phenotypes and 3 genotypes. But here we have 3 phenotypes as well as 3 genotypes. Okay? And the phenotypic ratio is also 1:2:1. The phenotypic ratio is also 1:2:1, and the genotypic ratio is also 1:2:1. What you are getting? Number of genotypes equal to number of phenotypes here. So obviously there is a deviation from the Mendel's monohybrid cross. Here the number of genotypes are 3 and the number of phenotypes are also 3. So this is the second case where phenotypic ratio is equal to genotypic ratio after test cross. Here also the GR and PR are the same. Okay, so this is a deviation from the Mendel's law. Remember this beta, GR, PR same, 1:2:1, and the phenotypes are also 3, genotypes also 3. Now, codominance. Codominance is a phenomenon where it's different. Here, the mutated allele is not the recessive allele, but it is the functional allele which will form the functional protein, but that protein will be a changed or altered protein. Okay. Suppose, for example, codominance, I am taking the example of AB blood group. AB blood group. And in the AB blood group, these two genes, that is IA and IB, these two are the different forms of the same gene. These are different forms of same gene, but they do not show the relationship of dominance and recessiveness. Earlier, we used to say that the two forms of a same gene, one is dominant, one is recessive. But here, there is no such relationship. These are two forms of the same gene, but they do not show dominance or recessiveness, but they are co-dominant. They are co-dominant. When they are present together, they will show the equal expression. So what is codominance? When the two forms of a same gene are present together, they will show equal expression. No one is lesser than anyone. They will show equal expression. That is why we call it as codominance. Okay, and the best example of codominance is AB blood group in the ABO blood group system. And another example is MN blood group, which is another type of blood group system. Okay, and also Rowan color, like you can see in some of the cows, there are white patches and black patches. So that is also an example of codominance. And here also, remember beta, here also in codominance, the PR is equal to GR. That is 1:2:1. So how many cases we have now where PR is equals to GR? Test cross, incomplete dominance, and codominance. Okay. Let's move further to multiple allelism. Multiple allelism. I've just told you that generally there are two forms of a same gene, right? That means there are two different alleles of a same gene generally. But what is multiple allelism? Multiple allelism is occurrence of more than two alleles of a gene. Occurrence of more than two forms of a gene is multiple allelism. And example is ABO blood group system. Example of multiple allelism is ABO blood group system. What does it say? Let's see. Suppose, for instance, I have taken the blood group gene. This is the blood group gene. And during the course of evolution, this blood group gene has undergone mutation, and that too repeated mutations. This gene has undergone repeated mutations to form more than two forms. Now, what is the result? That in a population, we have how many forms of the same gene? We have, suppose I'm taking the example of blood group genes, we have three forms. And these three forms are IA, IB, and IO, also written as small I. IA, IB, IO. These are three alleles 3 forms of the same gene. Usually there are 2 alleles, 2 forms of the same gene. But in multiple allelism, a same gene has more than 2 forms or 2 alleles. Here you can see there are 3 forms of a same gene, that is the gene of blood group. This is multiple allelism. But what you have to remember here, beta, that multiple allelism is a trait of population. Multiple allelism is a trait of population. That means you cannot detect the occurrence of multiple allelism in an individual. You will have to take the support of population to understand this phenomenon. Like how we understand this, suppose my blood group is B. Suppose I have the blood group B. So suppose this, my blood group is this. Okay. And suppose my sister's blood group is AB. Right? So, and my daughter's blood group is suppose A. So in an individual, how many forms you can see? In an individual, you can see maximum 2 forms. Sorry. You can see maximum 2 forms. IA, IO, right? These are the 2 forms of a same gene in an individual. And minimum, how many forms you can see in a single individual? 1. So in a single individual, minimum you can see 1 copy of a gene, 1 form of a gene, and maximum in an individual, you can see 2 forms of a gene, not more than that. That means you will have to study the whole population to understand the phenomenon of multiple allelism, because at the max you can see only 2 different forms of a same gene in an individual. But if you want to see different forms, more than 2 forms of a gene in you'll have to take help of the population. This is suppose this is a population 1, 2, 3. How many forms you can see? 3. So this is how multiple allelism cannot be studied in an individual but in a population. And if you want to calculate the number of genotypes possible, if you want to calculate the number of genotypes possible if the number of alleles are given, then the formula can be n by 2 n plus 1. N is the total number of alleles. So suppose I am taking the example of blood group. So for calculating the number of genotype in blood group, I will write it as 3 by 2, 3 plus 1 comes out to be 6. That means in a population, total 6 genotypes are possible. And what are they? IA, O. These are the genotypes of a person with blood group A. Then IB, IO, B, and B, then IO, or simply I. Right? So how many blood groups are there? A, B, AB, O. And how many genotypes are possible? 6. So this is multiple allelism. Now let's understand what is pleiotropy. Pleiotropy, usually, let's take the usual example first. One gene controls one trait. Okay, one gene has a control over one trait or one expression. But when one gene controls several traits or several phenotypes at the same time, that phenomenon is called as pleiotropy. Effect of a single gene on multiple phenotypic expression. When a single gene is having effect on many phenotypic expressions, then that is pleiotropy. And the examples are gene of starch branching enzyme, the mutated gene in the disease phenylketonuria, and the mutated gene which causes the disease sickle cell anemia. All these are the pleiotropic genes. These are the examples of the pleiotropic genes. Let's discuss them in detail. Starch branching enzyme. Starch branching enzyme gene. NCERT say it as B gene. As B gene. Okay. This gene beta is responsible for the formation of starch grains. This is responsible for the formation of starch grains. Okay. But along with that, it also controls the seed shape. So this gene is not controlling only one trait, but it has effect over two traits. So this gene has effect on two traits. Effect on two traits. One is formation of starch grain or size of starch grain. And the other trait it is controlling is seed shape. So obviously it is a pleiotropic gene because it is having effect on two traits at the same point of time. Now let's see how this gene controls the starch formation. If this gene is in this form, these are the three forms which are possible. If this gene is in the heterozygous dominant form, then it will form the starch grain with large size. So, large starch grains. Are formed. But if this is present in the heterozygous condition, then it will form intermediate starch grains. And if it is present in the homozygous recessive state, then it will form small sized starch grains. Okay. So what is important here to understand that when this gene is controlling the formation of starch grains, then this gene is showing incomplete dominance. In controlling this trait, this gene is showing incomplete dominance. You can see here, capital B, small b should also be having the large size, but no, it is having the intermediate size. That means this is incomplete dominance. So this gene, when controls this trait, it shows incomplete dominance. Let's see here what is happening. If this gene is present in this form, then the shape of the seed is round. In this case also, the shape of the seed is round, and in this case is wrinkled. That means in controlling the seed shape, this gene shows complete dominance. So So this is important, that this gene, B gene, is a pleiotropic gene controlling two traits together, but in controlling this trait, it is showing incomplete dominance, and in controlling, while controlling this trait, this is showing complete dominance. Okay, so this is important to remember. Now let's take the example of another example of pleiotropy, that is phenylketonuria. Phenylketonuria is a disease in which there is a gene involved. That gene is phenylalanine hydroxylase enzyme gene. It's also called as PA gene. Okay. So beta, what happens during phenylketonuria? It's a genetic disorder. During this disease, this particular gene becomes mutated. So what happens in the body naturally? The amino acid phenylalanine usually gets converted into another amino acid, that is tyrosine, and this is done with the help of PAH enzyme, phenylalanine hydroxylase enzyme. And when this will be formed? When this gene is present in the dominant form. Obviously, when this gene is present in the dominant form, then this enzyme will be formed and there will be conversion of phenylalanine to tyrosine. But when this gene mutates, then it becomes recessive. And if it is recessive, then phenylalanine will not be converted to tyrosine. Why will it not be converted to tyrosine? Why? Because this enzyme, PAH enzyme, is not formed. Why it is not formed? Because the gene has become recessive or mutated. And this recessive or mutated allele, which is causing the disease phenylketonuria, this mutated allele is actually pleiotropic in nature. So during the disease phenylketonuria, this gene, PAH gene, mutates, become recessive, and actually this recessive allele is pleiotropic in nature. That means it will have multiple effects. And what are the multiple effects of this pleiotropic gene? This PAH mutated gene. That means the recessive allele, the recessive form. It is pleiotropic in nature and it is controlling many traits together. What is the primary effect of this mutated allele? The primary effect is accumulation of phenylalanine. Accumulation of phenylalanine. Accumulation of phenylalanine in the form of phenylpyruvic acid. The form of phenylpyruvic acid. So generally, phenylalanine is converted into tyrosine, but in this disease, because the gene and forming this enzyme is recessive, so no enzyme will be formed. Phenylalanine will not be converted into tyrosine, but it will be accumulated in the form of phenyl pyruvic acid, and this can be detected in the urine also. And when this gets accumulated in neurons, it becomes fatal. And what is the secondary effect of this mutated gene? The secondary effect is mental retardation, and depigmentation in skin and hair. Okay. So this recessive or mutated allele which caused the disease phenylketonuria is actually pleiotropic in nature, causing these effects: mental retardation, depigmentation in skin and hair, and the accumulation of phenyl pyruvic acid. Now, the another example of pleiotropic gene is the HBS gene. So, for that purpose, we'll have to discuss sickle cell anemia. So, beta, you know that inside the RBCs, we have alpha chains and beta chains, which together forms the hemoglobin molecule. Sickle cell anemia is a disease where the beta chain of the hemoglobin gets defected. And how does that happen? Let's see. Sickle cell anemia. Suppose I'm taking the example of normal hemoglobin gene, that is HbA gene, right? Normal hemoglobin. In a normal hemoglobin gene, I'm taking this, this is the DNA. This is the DNA, and the whole gene which controls the formation of beta chain is a big gene, right? And I am taking only one codon of that big gene where the change occurs. Okay, the gene which is present on DNA, which is responsible for the formation of a beta chain, I'm taking only that codon of this gene where the change will occur. So let's focus on that codon. So in the normal case, this is the template strand. Now you know what is a template strand and a codon strand. Okay. On DNA, transcription will occur and mRNA will be formed. This mRNA will be having the codon GAG, right? The mRNA has the same sequence of the coding strand and the complementary sequence as of the template strand. This GAG, when undergoes translation, it forms the amino acid glutamic acid. But when this gene mutates, then the mutated allele is the HBS form. HBS. So this is the mutated form of the normal gene, that is HbA gene. And what happens during mutation in this gene? There is a change. See the change. See the change. This is the template strand on which mRNA will be formed. On this template strand, this thymine is replaced by adenine. Thymine is replaced by adenine. This is a case of point mutation. Point mutation means the mutation has occurred only at one point. So when mRNA will be formed over this mutated allele or mutated gene, then the codon formed here will be GUG, and the amino acid ultimately formed will be valine. Valine. Okay. So this mutated gene, what's the mutation? That at a particular point, at this particular point in a codon, thymine is changed, is substituted by adenine, and initially glutamic acid used to be formed, but here valine will be formed. So what will happen? So you can say that HBS is a pleiotropic gene. The pleiotropic gene. And what is the primary effect of this pleiotropic gene? Primary effect is that in the normal condition, in the beta chain of the hemoglobin at 6th position, the amino acid is glutamic acid. But in the beta chain at the 6th position, now valine will come. So what is the primary effect? Primary effect is substitution of glutamic acid by valine at 6th position of beta chain, which affects the binding capacity of hemoglobin with oxygen, and it changes the shape of the RBCs. So what are the secondary effects? The secondary effects are change in shape of RBC, change in shape of RBC, it becomes biconcave, then reduced oxygen transport, physical weakness, and resistance towards malaria. Okay. So that is why HBS is a pleiotropic gene, because it has several effects on the phenotype. This change, glutamic acid replaced by valine, and these are the secondary effects. So there are some terms which are important regarding sickle cell anemia. What are those points? That pleiotropic effect is seen by the HBS. It's an example of pleiotropy, and HBS gene is the pleiotropic gene over here. Sickle cell anemia is a case of mutation which is called as a substitution, and the substitution is done at a single point. So it is a case of point. These are the terms related with sickle cell anemia. Pleiotropy, substitution, mutation is a kind of substitution, and point mutation. And it is a autosomal recessive disorder. That means the gene which is responsible for sickle cell anemia, that is the HBS gene, is present on a autosome. So these are some terms which are related to sickle cell. Now let's move to the another phenomenon, that is called as polygenic inheritance. Polygenic inheritance. Usually one trait is controlled by one gene, but the name is suggesting polygenic inheritance. That means here one single trait is controlled by many genes. Opposite of pleiotropy. Pleiotropy, what was happening in pleiotropy? One single gene was having effect on several phenotypes. But here, one expression, one phenotype, one trait is controlled by many genes. So polygenic inheritance. And in another words, it is also called as quantitative inheritance. Also called as quantitative inheritance. Okay, that means here the quantity of the genes will decide the intensity of the expression. Mendelian inheritance is qualitative inheritance. There was no effect on the phenotype by the number of dominant alleles. In the Mendelian inheritance, capital T was also 10 centimeter tall. Capital T, small t was also 10 centimeter tall. So it is not the number of dominant alleles which was affecting the expression, but the presence or absence of dominant allele which was affecting the expression. So Mendelian inheritance is the qualitative inheritance where number of dominant alleles doesn't work. The presence or absence of dominant allele is important. But in polygenic inheritance, the number of dominant alleles are responsible for the intensity of the expression. The more the number of dominant alleles, the more would be the intensity of expression. Now we will understand what is the meaning of this point. What is saying that here one trait is controlled by many genes, and where you can see this polygenic inheritance, where you do not have only two choices. Like in the Mendelian inheritance, we had only two choices, either tall or dwarf. No, these traits are seen where you cannot very distinguish between the expression. They are present across a gradient. That means, suppose skin color inheritance, there are not just the two options that either very white or either very black. No, there are so many skin tones, right? So this trait is not distinct, but this trait is present across the gradient. Mane, there are various forms of this trait. You cannot say that one person is genius like Einstein and one person is very duffer like Donkey. No, there are so many persons with different IQ values. So, and likewise, height in humans, you cannot say that there would be only two kind of people in the population, 6 feet tall or 5 feet tall. No, there are so many, varieties. So polygenic inheritance is seen where the traits are present in the gradient. So height in humans, skin color in human, or intelligence in humans. These are the examples of the polygenic inheritance. And here each dominant allele contributes to the expression. Each, because here the only presence is not enough. Number of dominant alleles. The more the number of dominant alleles, the more would be the expression. So effect of each allele is counted. So effect of each allele is additive. Okay. So let's understand the polygenic inheritance more with the example of skin color inheritance. Skin color inheritance is controlled by 3 pair of polygenes. controlled by 3-pair polygenes. That is A, B, C. 3 genes together, they control the skin color. And capital A, capital A, capital B, capital C, capital C. All the alleles are dominant. All the 6 alleles are dominant. That means this person would be Negro or very dark, right? Very dark. And this person has the 0 dominant allele, 0 dominant allele. So it would be albino or very light. Okay. So yes, there is effect of, now see how the effect of dominant allele is there. Number of dominant alleles is there. So when we cross them and, you know, like a selfing, it seems like a trihybrid cross. Selfing is done, then in the F1 generation, these kind of individuals will come. And then in the F2 generation, we'll be having many options. We will be having many options. And what are they? Let's discuss. There would be individuals with 6 dominant. That is A, B, B, right? And these individuals, the phenotype of these individuals would be Negro or very dark. Okay. Then if we reduce the number of dominant alleles, 5 dominant alleles, there can be various genotypes with 5 dominant like this. How many dominant alleles here? 1, 2, 3, 4, 5. And you can form various genotypes having 5 dominant alleles. Like this can also be the case. This also have 5 dominant alleles. So make the genotypes yourself in your copies, that how many possibilities can be there with 5 dominant alleles. And if the dominant alleles are 5, then the expression would be dark. Then as we go on reducing the number of dominant allele, the expression, the intensity of the expression will reduce. If there are 3 dominant alleles, then it would be intermediate condition. Then if 2 dominant allele, 1 dominant allele or 0 dominant allele, then the condition will change to 1 or 0. It would be fairly light, or very light. Okay, so see how many phenotypes are possible. As you are decreasing the number of dominant alleles, the expression is getting reduced. That means each dominant allele is participating in reducing the expression. And as you are reducing the dominant allele, the expression will reduce. Okay? These individuals, as it is it's a type of trihybrid cross, so total type of combinations possible here would be 64, and out of them the possibility of this can be 1, of this can be 6, of this can be 15, of this can be 20, again 15, 6 or 1. That means the ratio you get from here, the ratio, phenotypic ratio you get would be 1:6:15 is 20 to 15 is to 1. And if you draw a graph between the number of dominant alleles, the number of alleles and the number of individuals, then it comes out to be a bell-shaped graph in which the maximum number of individuals are intermediate individuals. And this kind of intermediate individuals with 3 dominant allele, they are called as the mulattoes. Okay, so this is polygenic inheritance. Now let's discuss the chromosomal theory of inheritance. In the 1900, after the year 1900, many important things were there. There was rediscovery of Mendel's work in the year 1900, and during 1902 and around it, there was advancement in electron microscopy. So laws of inheritance were there in the picture, and the electron microscopic studies, because of the help of electron microscope, we had studied the dividing cell under the microscope in which meiosis was occurring. So we studied the movement of chromosomes during meiosis, during division, with the help of electron microscope. So and around 1902, we had laws of inheritance, and we had the proof of the law of inheritance that yes, there is movement of chromosomes, right? So now we have the knowledge of chromosomes. How do they move? How do they segregate during the anaphase? Now we have this proof, and the basis of this knowledge, Sutton and Boveri, they combined the knowledge of the electron microscopic studies and the laws of inheritance, and they concluded that the behavior of chromosome is parallel to the behavior of factor or gene. The behavior of chromosome is parallel to the behavior of gene. And these are based on rediscovery of Mendel's work and the advancement the electron microscopy. Now, what is the meaning when I say that there is parallelism between the behavior of gene and chromosome? Like both of them, chromosomes as well as gene, they occur in pairs in a diploid individual. Same thing. During meiosis, during gamete formation, homologous chromosomes also segregate and the genes also segregate. And during fertilization, the paired condition of chromosomes gets restored, as well as the paired condition of genes also gets restored. So the gene and the chromosomes, they show the same behavior. Actually, the Sutton and Boveri together, they combined these studies, but the chromosomal theory of inheritance was actually proposed by Sutton. There is a line in NCERT, Sutton proposed the chromosomal theory of inheritance. So the knowledge of Mendel's work in electron microscopy were combined by Sutton and Boveri, but the chromosomal theory of inheritance was given by Sutton. Now this is a table given in NCERT, and this is pretty important. This says that now we have to tell that what is A and what is B here. Let's understand. A occur in pairs. Yes. B also occur in pairs. Beta, this A column represents, this B column represents chromosome. You must have studied it the opposite way, but the correct version is this, gene and chromosome. Okay, how? See. Both occur in pairs? Yes. Both segregate at the time of gamete formation? Yes. Now understand. Independent pairs segregate independent of each other, and one pair segregates independently of another pair. See, this line, one pair segregates independently of another pair, is not cannot be universally applied for gene. Remember, gene, it's not possible that one pair of gene is always independent of other pair of gene. It is not always possible, because the genes present on the same chromosomes, in that condition, one pair of gene is not independent of the other pair of gene, right? The genes present on the same chromosome, they show linkage, and in that condition, one pair cannot be, free of the other pair, independent of the other pair. So this line cannot be usually applied for gene. But yes, in case of chromosomes, one pair of chromosome is always independent of the other pair of chromosomes, right? Independent pairs segregate independently of each other. This is correct. Independent pairs means the genes which are present on different chromosomes. Only independent pairs can segregate independently of each other. And what are these independent pairs? Independent pairs are the genes present on the different chromosomes. Okay. So let's move on to the phenomenon of linkage now. What is linkage? Linkage is a phenomenon which tells us about the physical association of the genes present on the same chromosome. The genes which are present on same chromosomes are said to be linked. And why they are said to be linked? Because they will always inherit together. They will always move together, right? So these genes represents a single unit which will always be inherited together. So they all are associated with each other and they all are linked. And this phenomenon is called as linkage. Now, to understand linkage, Thomas Hunt Morgan, who is called as the father of experimental genetics, he carried out various dihybrid crosses in the organism Drosophila melanogaster, which is commonly called as fruit fly. Okay. Now let's understand what he did. To understand that, first of all, let me take some examples. Let me take three cases, okay? I'm taking two genes, right? I am taking two genes. And these are present on the different chromosome. And if these two genes are present on the different chromosome, they will show independent. And we will have, from here, capital R can come, from there capital Y can come, here small r, capital Y, capital R, small r, small y. Probability of all the cases is 25%, 25%, 25%, right? These two are the parental types. Why parental types? Just capital. Why parental types? Because you know what parents we took in the Mendel cross? Capital R, capital R, capital. Okay. And these two forms are the recombinant forms. So what you can see here, beta, that when independent assortment is there, and when there is independent assortment, the genes are present in different chromosomes. So when independent assortment is there, you will be having 50% parental and 50% recombinant, right? This is the normal case. Let's take the second case. In the second case, I'm saying that the genes are present on the same chromosome. are present in the same chromosome, and if they are present on the same chromosome, so linkage. So linkage, independent assortment will not be there. This linkage can be of two types. It can be complete linkage or it can be incomplete. In complete linkage, there is close association between the two genes. Close association. They are present very close to each other, and there is no chance that they will be away from each other. They love each other so much that they will always be together. So there will be no formation of recombinants. If the linkage is complete, then there would only be 100% parental. But if there is incomplete linkage, then the genes are present on the same chromosome but a bit far away. That means they love each other, but sometimes they gets angry with each other and they can go away from each other. So these two are present on the same chromosome, but they are present at a distance. So there are chances that this gene can be separated from this gene. And how this can happen? With the help of an agency called as crossing over. You know what is crossing over? Exchange of segments during the Pectine stage of meiosis 1. So there is a chance that during crossing over, this capital Y goes away and they can be separated. So there is linkage, but there are chances that they can be separated with the help of crossing over, and that is incomplete linkage. In incomplete linkage, the parental types will be more than 50%. More than 50%. And the recombinants will be less than 50%. Okay. So in the case of independent assortment, 50% parental, 50% recombinants. In case of complete linkage, no recombinants, 100% parental. But in case of incomplete linkage, because of crossing over, there are chances that recombinants can be formed, but they will be less than 50%. Okay. So now we have understood this thing. Let's understand the experiment of the Morgan. But before understanding this, let's understand that what are the traits that Morgan took for his experiment. Morgan took sex-linked trait for his experiment. Sex-linked traits. Sex-linked traits are those traits whose genes are present only on X or Y chromosome, are present on sex chromosome. That means these genes are not present on the autosomes, but they are present on the allosomes, that is sex chromosomes. They can be present either on the X chromosome or on the Y chromosome. So all those traits whose genes are present either on the X chromosome or on the Y chromosomes, they are called as the sex-linked traits. Now Drosophila is also a diploid organism having 4 pairs of chromosomes. That means total 8 chromosomes, and males are 6 plus XY and females are 6 plus XX, right? Males are XY, females are XX. So Morgan took 3 traits for his experiment, and these 3 traits, beta, remember, they are X-linked traits. These 3 traits are X-linked traits. That means the 3 traits taken by the Morgan, they are X-linked traits. That means the genes of all the 3 traits taken by him are present only on X chromosome and nowhere. So let's take the example of those 3 traits. Those 3 traits were eye color gene, body color, and wing type and wing. Okay, eye color can be red or can be white. Body color can be yellow, brown or yellow. Wing type can be normal, where the wing is normal, or miniature, where the wing is vestigial or not properly developed. Okay. So all these 3 traits, the genes are present only on the X chromosome. Now there, how we can denote the dominant and recessive allele? We know that the dominant allele is represented by a capital letter and the recessive allele is represented by a small letter. But there are another ways also. Dominant allele can be simply written as plus, or it can be written as small symbol plus. So these are the 3 ways to represent a dominant allele: capital T or simple plus, or small symbol and plus over it. Okay. So for white eye color, which is a recessive trait, I am taking the small symbol, that is small w, and for red I am taking small w plus. So small w plus is showing the dominant gene. Here small y and y plus. Brown is the dominant trait, y plus, yellow is the recessive. Miniature, small m and m plus. Okay? Clear? Now let's move on to his experiment. He did 2 experiments which are given in, actually he did 3 experiments, but in NCERT 2 are given. So let's discuss them. For first experiment, he took eye color and body color. What he did, he took the parents. Okay, he took the parents. The female has 2 X chromosome, one this, one this. The female taken by him was recessive. Recessive means it would be yellow bodied and white eyed, right? Yellow bodied, white eyed. So what symbols should I write over here? Small yw, small yw, because this female is a recessive female. He crossed this recessive female with the male, and male has X and Y chromosome. Has X and Y chromosome, and you know that the gene of eye color and body color are not present on Y chromosome. So we will not draw them on Y chromosome. So the male he took was a dominant male. So Y positive, W positive. So male was wild. wild or dominant, and that means it is red-eyed and brown-bodied. Okay, red-eyed and brown-bodied. So female taken by him was recessive and the male taken by him was dominant. Now, what kind of gamete it can form? The female, it can form only one type of gamete, and that is only this, small y w x. But male can form two types of gametes. That is, in one condition, Y chromosome can come, and in the another one, the X chromosome will go. The X chromosome will go and it will have Y positive, W positive. Okay, and then if we will cross these gametes, then what we will get? We will get females X cross with Y, X with Y, we will have male, and X with X, we will have female. Right? So these are the offsprings of these parents. These are the males. Now males have become recessive and the females have become dominant. But what kind of dominant? Heterozygous dominant. What kind of dominant? Heterozygous dominant. Now, this male, if we do selfing of them, if we cross them together, this male can give two types of gametes. In one, X will go, and in other one, Y will go. This female can produce four types of gametes, that is Y, W, Y positive, W positive, Y, W positive, or W, Y positive. So male can give two types of gametes, female can give four type of gametes, right? So offspring would be how many types? Eight types. Okay, so let's see in the diagram given here. These are the parental types. These four are the, out of 8, these 4 are the parental types and these 4 are the recombinant types. Okay, these 4 are the recombinant types. What are parental types, beta? In all the combinations where Y is present with W and Y positive is present with W positive, in all those combinations, they would be the parental combination. But those combinations where Y positive is with W and Y is with W positive, these are the recombinant combinations. So out of 4 offsprings in the F2 generation, out of 8 offsprings in the F2 generation, 4 showed the parental combination and 4 showed the recombinant combination, right? But if it would have been an independent assortment, then 50-50 ratio would have been there. But here, see what's happening here. The parental type are 98.7% and the recombinants are 1.3%. See what I've just told you, that in complete linkage, 100% are parental types. But if there is incomplete linkage, then recombinants are less than 50% and parental are more than 50%. Here the parental are 98.7 and the recombinants are 1.3%. That means there is incomplete linkage. And when he took the eye color gene and body color gene, then the recombinants were 1.3%. But when he took the eye color gene and wing type gene, then the recombinants were 37.2% and the parental combinations were 62%. Getting the point? So he did two sets of experiment. Eye color, body color, these are the parental combinations. Eye color, wing type, these are the parental combinations. So what we understood from this experiment, that there is more linkage. There is more linkage in between the genes eye color and body color, because here the number of recombinants are just 1.3%. But eye color and wing type has linkage, but the linkage is lesser than the eye color and body color, because here the recombinants are 37.2%. So what is the conclusion? What is the conclusion over here? And yes, beta, the question directly comes when Morgan took eye color and body color gene, what were the number of, what were the proportion of recombinants? You just have to learn this, okay? Eye color, body color, 1.3% recombinant. Eye color, wing type, 37.2% recombinant. And you will calculate the parental type by, subtracting it. Just learn 1.3% and 37.2%. Eye color, body color, eye color, wing type. Question directly comes. Okay, so what is the conclusion? Conclusion says that distance between two genes, present on same chromosome, is inversely proportional to linkage. That means the more the distance between the genes on the same chromosome, the less would be the linkage, and the more closer they are, the more is the linkage. And linkage is inversely proportional to crossing over, because distance between genes present on same chromosome is directly proportional to crossing over. The more they are distant from each other, the more will be the chances of crossing over. That means where there is linkage, where there is more linkage, there are less chances of crossing over, and where there is crossing over, there are less intensity of linkage. Okay, so this was the conclusion of Morgan's experiment. So this says that the eye color gene and the body color gene are present closer to each other, and the linkage is more tighter, and the eye color and wing type gene, they are present farther to each other, and the linkage is a bit lesser. Okay. Now, recombination and crossing over. Recombination and crossing over is the agency. It is the tool with which the linked genes can be separated and they can show recombination, right? Because we know what happens during crossing over. There is genetic exchange. There is exchange of arms. Exchange of arms between non-sister chromatids. These are the sister chromatids of the same chromosome, and these are the non-sister chromatids. Exchange of arms between non-sister chromatids of homologous chromosomes. Where it occurs? It occurs at packaging of meiosis 1. Okay. There is genetic change, and this is called as crossing over. And what is the result of crossing over? Recombination, that is formation of new combination. Okay. Now, Alfred Sturtevant was the student of Morgan. He was working with Morgan on Drosophila, and he deduced the term gene mapping or chromosomal mapping, he understood one thing. If we know the number of recombinants in a population, then with the help of that number, we can have an idea that the genes present on the chromosome are closer or farther. That means we can predict the position of genes on the chromosome with the help of the recombination frequency or with the help of the number of recombinants present in a population. So he talked about the recombination frequency. He talked about the recombination frequency, which is also called as COV or crossover value. Which is equal to number of recombinants, total number of recombinants in a population upon total number of progeny to 100. If we know that the number of recombinants are, how many recombinants are there in a population, then we can predict the recombination frequency or crossover value. And this crossover value is equal to the distance, present between genes on the same chromosome. Okay. Suppose, for instance, I am saying that the recombination frequency between two genes is 20%. The recombination frequency or crossover value is 20%. Then what is the meaning of this? That means the distance between the two genes is 20 map units or 20 centimorgan. The distance which we use for the calculation of distance between the genes on chromosome are either measured in the form of map unit, or in the form of centimorgan. Right? Okay, if I say that the recombination frequency is 60%, then what would be the distance? Obviously, no, if I say that the distance between two genes present on the same chromosome is 60 map unit, then what would be the recombination frequency? Obviously, the recombination frequency will not be 60% because the maximum recombination frequency can be 50% even when the genes are present on different chromosomes. Okay, but you can predict with the help of recombination frequency that what can be the distance between the two genes on the chromosome. And how you define one map unit? One map unit means there are 1% chances of crossing over between the genes and 1% recombinants can be seen in the population. Okay. Now let's move on to the other part of the chapter, that is genetic disorders. These are the congenital disorders. That means you cannot correct them. They are present by birth. Now, genetic disorders are of two types. They can be Mendelian disorders and they can be chromosomal disorders. Remember, yeah, Mendelian disorders are gene-related disorders. They are gene-related disorders, and the chromosomal disorders are chromosome-related. And what is the cause of Mendelian disorders? Cause is change in sequence of gene. Change in sequence of gene. When there would be any change in the sequence of gene, then it would be a Mendelian disorder or gene-related disorder. But if the cause But if the cause is either change in number of chromosomes, either change in number of chromosomes or change in structure of chromosome. Okay, remember this. Mendelian disorder, change in sequence of gene, chromosomal disorders or non-Mendelian disorders. These are also called as non-Mendelian disorders. The change will be in the structure of chromosome. That means suppose one arm of the chromosome is lost or it is added with extra arm. So if there can be change in the structure of chromosome or there can be change in the number of chromosome. The number of chromosome can be increased or decreased. That would be a chromosomal disorder. Okay, so this kind of questions are very common. There is a list of diseases in a box and it is asked that how many of them are Mendelian disorder and how many of them are non-Mendelian disorders. So sickle cell anemia, phenylketonuria, hemophilia, color blindness, thalassemia, these all are Mendelian disorders. And Down syndrome, Turner syndrome, Klinefelter syndrome, these are non-Mendelian or chromosomal disorders. So let's discuss them one by one. Color blindness. Color blindness is a sex-linked trait. Now you understand the meaning of sex-linked inheritance. That means the gene of color blindness is present on X chromosome. Present on X chromosome, right? On X chromosome. And color blindness doesn't mean that your life is black and white. No, color blindness is generally red or green blindness. That means the cone cells of eye, they are unable to perceive the red and the green color. Okay, why it happens? Because the gene present on the X chromosome becomes recessive. It is unable to form the pigments and the cone cells are unable to perceive the red and green color. Okay, color blindness is more common in males than in females. In males, there are chances of about 8% chances in a population of color blindness, while in females, there are just 0.4% chances. Okay, this is a data given in NCERT. This is important. And why this is so? Yes, this is so because for a female to be color blind, both the chromosomes, both the X chromosomes should have the mutated recessive allele. This is a diseased female. Okay, but in a male, this is a diseased female, but in a male, only one copy of the recessive allele is enough for causing the disease. So this question often comes that why is it so that the color blindness is seen more in males as compared to females? You will say the answer will be because in a female, the disease will be caused only when there would be two copies of the recessive allele present on both the X chromosomes. But in males, only one copy will do because this is not found on the Y chromosome. Right? Okay. And the question comes from color blindness like this. If let's do one question. If a female had a colorblind father, if a female had a colorblind father, then no, colorblind father and marries a, this female marries a colorblind man. The colorblind man, then you have to calculate one. proportion of diseased progeny. B, proportion of normal males. And C, proportion of diseased females. Okay. If a female had a colorblind father, this female marries a colorblind man, then you have to calculate these things. So we know that a female had a colorblind father. If the female had colorblind father, that means this female would be a carrier, because in a female, one X chromosome comes from mother and the other X chromosome comes from the father. That means this female is a carrier female. The carrier. Carriers are normal. They do not show color blindness, but yes, they carry the gene for color blindness. Marries a color blind male. So what will be the conditions? Okay, so you can say these are the progeny, right? This is the carrier female, which is the normal female, normal male, diseased female, and diseased male. Now they are saying we have to calculate the proportion of diseased progeny. The proportion of the diseased progeny, total number of progeny are 4. Okay, and how many are diseased? 2. So comes out to be 50%. Proportion of normal males. Males are, probability of the child being a male is 50%, and out of that only how many chances are there? See here, one of the chances you can see here of male can be of colorblind male, and other chances the normal male. So 1 by 2 chances are there, that is 50%. Then proportion of diseased female, proportion of diseased female, out of 2 the chances are of only 1, that is 50%. So remember this, when progeny is asked, denominator mein you will write 4, but when the males are asked, denominator, in the denominator you will write 2, and when females are asked, in the denominator you will write 2. Okay, so this is how you will solve the question of color blindness. Now hemophilia. Yes, and one more thing that I have just told you, that it is present on X chromosome. So color blindness is a X-linked recessive disorder. It is X-linked recessive disorder. Okay. Hemophilia is also a sex-linked trait, because the gene causing hemophilia is also present on X chromosome. So this is also a X-linked recessive disease, just like color blindness. X-linked recessive disease. This is also called as the royal disease because it was present in the pedigree of Queen Victoria. And what happens during this disease? Blood clotting is absent. As soon as there is a cut at any body part, the blood, as soon as it comes in contact with the air, blood clotting is there. But in the hemophilia, a simple cut can cause the death of the person, because the bleeding will not stop, because blood clotting is absent. So because the gene has become recessive, so any of the clotting factor, you know that blood clotting is a cascade effect where many clotting factors are involved. If any of the clotting factor is not formed, then the blood clotting will be absent. And hemophilia further can be of two types. It can be hemophilia A and hemophilia B. Hemophilia A is present when the clotting factor 8 is not formed. Clotting factor 8 is not formed. And what is that clotting factor 8? The name of that clotting factor 8 is anti-hemophilic globulin. Anti-hemophilic globulin. Hemophilia B occurs when clotting factor 9 is absent. Clotting factor 9 is absent. And clotting factor 9 is also called as the Christmas factor. It's also called as the Christmas factor. Okay? So this is hemophilia. Then comes the disease thalassemia. And the questions of hemophilia will be same as the questions of color blindness. They will be solved in a similar way because both of them are X-linked recessive traits. And yes, all the X-linked recessive traits, they show crisscross inheritance. What is crisscross inheritance? The passage, the transmission of characters is through the opposite sex. Like the female will transfer this gene to her granddaughter through her son. This is called as crisscross inheritance. And all the X-linked recessive traits like hemophilia and color blindness, they show crisscross inheritance. Now comes the thalassemia. Thalassemia is also a Mendelian disorder, but it is not a sex-linked trait or X-linked trait, but it is an autosomal recessive disorder. That means it occurs when a gene becomes recessive on an autosome. What happens in thalassemia? We know that in RBCs, alpha chains and beta chains together, they form hemoglobin. Now think, if one of the chains are not formed or they are formed in a very low quantity, so the polymerization of alpha and beta chains will not be normal and the RBCs will rupture after a point of time, and this is called as thalassemia. And the patients which are affected by thalassemia, they need a regular transfusion of blood. Regular. If it is severe, then every 20 days they need blood transfusion. Okay, so it is formed due to reduced synthesis or absence of synthesis of one of the globin chains. So based on that, thalassemia is of two types, alpha thalassemia and beta thalassemia. In alpha thalassemia, there is reduced or absence of synthesis of alpha chain. Alpha chain will not be formed. And what is the reason behind this? Important. Learn with me. Important. There is a deletion in the two closely linked genes, two genes which are present on the same chromosome, that is chromosome 16. And the name of the genes are HBA1 and HBA2. Learn with me. Alpha thalassemia, that means reduced synthesis or no synthesis of alpha chain, and the reason is deletion in two closely linked genes, that is HBA1 and HBA2 present on the chromosome number 16. Do not get confused. Alpha 16, okay? In beta thalassemia, there would be reduced synthesis of beta chains. The beta chains will not be formed or will be formed in very low amount, and the reason for this is deletion in the gene, and the name of the gene is HBB gene present on chromosome number 11. Okay, do not get confused, beta. Beta 11, alpha 16, HBA1, HBA2, HBB gene. Okay, so this is thalassemia. Now comes, so Mendelian disorders are finished. Now discuss the non-Mendelian or the chromosomal disorders. Here comes the Down's syndrome. What is the reason of Down syndrome? Trisomy of 21st pair of chromosomes. Okay, and what is trisomy? That usually we know that 23, we have 23 pairs of chromosome. That means in a pair 2 chromosomes are there. But here in case of Down syndrome, the number of chromosomes increases from 46, it becomes 47, and in the 21st pair there are not 2 chromosomes, there are 3 chromosomes, and this is called as trisomy. Okay, now there is trisomy of 21st pair of chromosome. And what is the cause of this sort of defect? The cause is beta non-disjunction of homologous chromosome. Non-disjunction of homologous chromosomes or chromatids during gamete formation. This is important. Non-disjunction. We know beta that in the anaphase 1 and anaphase 2, during anaphase 1 there is separation or segregation or disjunction of homologous chromosomes, and then ultimately in anaphase 2 there is disjunction or separation of chromatids. But if this segregation, this disjunction do not happen, then there would be one extra chromosome in one gamete, and in one gamete the chromosome will not go, right? So in one gamete there would be, if suppose there in gamete there are 23 chromosomes, but 24 chromosomes will be there, and in one chromosome there would be 22. So the reason of the diseases like Down syndrome is the non-disjunction. That means there would not be the separation or segregation of homologous chromosomes or chromatids. This is very important. Non-disjunction of chromatids or homologous chromosomes during gamete formation. Suppose for instance, during anaphase, every chromosome, pair of chromosome are aligned like this, you know, all the pairs are aligned like this. So in this first pair, everything is normal. One chromosome this side, one chromosome this side. But suppose in 21st pair, both the chromosomes go at one pole, and on the other pole, nothing goes. So in every pair, things are going normal. One this side, one this side, one this side, one this side. But in the 21st pair, both the chromosomes goes to one side, and to other side no one goes. That means what will be the situation? One gamete will have the condition n+1, and the other gamete will be having the condition n-1. N-1, right? N is 23. So here the number of chromosomes will be 24, and here the number of chromosomes will be 22, because there is, this pair has shown non-disjunction or non-separation of the chromosomes, right? And when this abnormal gamete, when this abnormal gamete fuses with the normal gamete, abnormal gamete is n+1 and the normal gamete is n. So what will be the situation? 2n plus 1. 2n plus 1. A normal human being is 2n diploid, right, with 46 chromosomes. But an individual with Down syndrome is 2n plus 1. That means 47. This individual has 47 chromosomes. That is, there is 2n+1 condition. And what is the reason behind it? You know it now. The reason behind is non-disjunction, and the reason is that there is fusion of abnormal gamete with the normal gamete, and thus 2n+1 condition arises, and that is Down syndrome. Okay. Now, these are some symptoms of the Down syndrome. The affected individual have short stature, the height is short, small round head, furrowed tongue, that means there is some depression in the tongue, then partially open mouth, the mouth is always partially open, and palm is broad with characteristic palm crease in the middle, which divides the palm into two equal halves, and physical, psychomotor and mental development is retarded. All sorts of developments are retarded. This is the diagram shown in NCERT. Some other symptoms: flat back of head, many loops on fingertips, palm crease, broad flat face, congenital heart disease. These are the additional symptoms of a person with Down syndrome. And here you can see, beta, this is the karyotype. This is the karyotype. Karyotype is the arrangement of the chromosomes according to the size. And if you see this karyotype, you can see something here. See carefully. You can see something here. Here. Every pair has two chromosomes, two. But this 21st pair, has 1, 2, 3 chromosomes. So this is the trisomy of 21st pair of chromosomes. Okay. Now let's discuss the Klinefelter syndrome. Klinefelter syndrome is seen in males. It is seen in males. And again, the example is same as that of Down syndrome. But what happens here? In Down syndrome, there was a trisomy of 21st chromosome. But in Klinefelter syndrome, there is a trisomy of the sex chromosomes. The last pair, the 23rd pair. Usually in a male, what we have 44 autosomes and 2 sex chromosomes, that is XY. So a normal male has 44 plus XY condition, right? But you can see that a male affected with the Klinefelter syndrome has 2X chromosomes. So there is trisomy of sex chromosomes here. And there is another way we can write this condition. The total number of chromosomes, either you write it as 44 plus XXY, or you write it as total number of chromosomes now increased 47, comma, what is the cause? XXY. So there are two ways to show the Klinefelter syndrome: 44 plus XXY or 47, XXY, because the total number of chromosomes now increased to 47. Okay. Now, because of this extra X chromosome in the individuals, what happens? There is a condition called as gynecomastia. Gynecomastia is development of feminine characters, that is development of breasts and other things. So in these kind of males, there are feminine characters present because of the extra X chromosome. And what is the reason behind it? There can be two reasons. First, either the abnormal egg was there. Abnormal egg will be 22 plus XX, that represents the 2n plus 1, sorry, that represents the n plus 1 condition. Okay, that, oh, just a second, beta. I'm a bit tired. Abnormal egg would be n plus 1. N plus 1. Okay. So, a normal egg has 23 chromosomes, but an abnormal egg would be having 24 chromosomes, and that would be 22 plus XX. A normal egg should have 22 plus X condition, but the abnormal egg has 22 plus XX condition, because there would have been non-disjunction between the X chromosomes. Both the X chromosomes came in the came on the one side, and on the other side no X chromosome. So because of the non-disjunction in the X chromosomes, here the abnormal egg 22 plus XX is formed, and the normal sperm. So when normal sperm fuses with the abnormal egg, normal sperm is 22 plus Y. So what will be the result? 44 plus XXY. Okay? Or in another case, the egg can be normal egg, or the abnormal sperm. Normal egg will be 22 plus X, but the abnormal sperm will be 22 plus XY. Right? Non-disjunction again. So 44 plus XXY. Okay? So this is how Klinefelter syndrome happens. Now, Turner syndrome. And yes, the males affected with Klinefelter syndrome are. Now, Turner syndrome is seen in females, and these females are also sterile, but this is the case of monosomy. This is the case of monosomy. Down syndrome and Klinefelter syndrome were the cases of trisomy, but this is the case of monosomy. That means in a normal female, the condition is 44 plus XX, right? But here you can see the condition is 44 plus XO. That means in the last pair, instead of 2 X chromosomes, there is only 1 X chromosome. So in the last pair, there is only 1 chromosome, and that is why it is called as monosomy. And in what way we can represent it? 45, XO. Total number of chromosomes 45 now, and why? Because of the XO condition. The females affected with Turner syndrome, they have short height, short stature, they are sterile, they have rudimentary ovaries. Rudimentary ovaries means ovaries are not fully developed, and other secondary sexual characters are also not developed. Other secondary sexual characters are also not developed. Okay. And reason again, same, abnormal egg. Reason is same, non-disjunction of chromosomes, abnormal egg, normal sperm. Abnormal egg here will be taking is n minus 1. N minus 1. The gamete in which X chromosome did not came. So n minus 1. That means 22 plus O, which does not have any sex chromosome. And normal sperm would be 22 plus X. Right? Normal sperm can be of two types, 22 plus X and 22 plus Y. So if the normal sperm is 22 plus X and the abnormal egg is this, then the female would be 44 plus XO. Or you can take the opposite case. Normal egg, that is N, and abnormal sperm, that is N minus 1, that means 22 plus O. Okay, so this is Turner's syndrome. So we are done with the genetic disorders now. Now the last part, that is sex determination. Yes, sex determination. That means in any individual, how the gender of the progeny is decided. The gender of the progeny can be decided either by environment or by specific chromosomes. Environmental sex determination, that means it is decided after the fertilization, after the conception, by the environmental factors like temperature. In some reptiles and in some animals like Crypadilla, the environmental conditions like temperature, they decide what would be the sex of the progeny. Chromosomal basis of sex determination says that there are certain chromosomes which decides the sex of the progeny, and that was first told by Henking. Henking was studying spermatogenesis in firefly, and during his studies he discovered some bodies called as X bodies, and today we call them as X chromosome, and he told that in 50% of the sperms of the firefly there were X bodies, and obviously they decide the sex of the progeny. So now we will discuss the chromosomal basis of sex determination. The chromosomal sex determination can be of two types, where there is male heterogamety and female heterogamety. Now what is heterogamety? Always remember beta that the sex of the progeny is decided by heterogametic parent. Is decided by heterogametic parent. Okay, that means let's take the example of humans. Females are 44 plus XX and males are 44 plus XY. Okay, that means the female can form only one type of gamete, that is X, in which there is only be X, 22 plus X, only this kind of gamete. But the males can form two different types of gamete, that is 22 plus X or 22 plus Y. That means the female will always form only one type of gamete, 22 plus X. But who is forming two types of gamete here? Male, that is 22 plus X and 22 plus Y. So who is heterogametic here? Male. And the sex of the progeny is always decided by the heterogametic parent. So initially in our society, when a female could not produce a baby boy, so it was suggested to the males that marry again. But they could not understand that it is not dependent on her. It is dependent on the male, that either X comes or Y comes, so that the baby would be girl or a boy. Okay, so heterogametic parent decides the sex of the progeny. So there are two cases where first case where males are heterogametic. This is the female which is homogametic, and this is the male which is heterogametic. XXXY, as it is found in case of humans and Drosophila. XX is the female and XO is the male. XO means there is only one sex chromosome. Okay, and here also the male is heterogametic, example grasshopper. In the female heterogamety, here the female decides the sex of the progeny and the female is heterogametic. Here ZZ is male and ZW is female. Here also ZZ, the homogametic parent, is male and ZO is the female. O means the female has only one sex chromosome. Now what sort of question can come from here? That in grasshopper the female can form only one type of gamete, that is A plus X, right? A represents the number of autosomes and X is the sex chromosome. But male can form two types of gamete, that is A plus X and A plus O, right? So a question can be asked that in which organism it is possible that one of the gametes or one of the sperms is devoid of sex chromosome. That means in one of the gametes, the sex chromosome will not be there. That would be the case of grasshopper. Here you can see 50% of the males do not have the sex chromosome. Likewise here, 50% of the eggs, 50% of egg will be A plus Z and 50% of the egg would be A plus O. That means 50% of the female gametes will not be having any sex chromosome, and that would be the example of butterfly. Okay, so let's see the case of sex determination in humans. Sex determination in humans is XXXY type. That means in our case, we have AA plus X females. That means AA stands for 44 and XX, this is the female, and AA plus XY, that is 44 plus XY. From here, we can get only one type of egg, that is 22X. But from here, we can have two types of sperms, that is 22 plus X plus Y. So there will always be, in every pregnancy, there will always be 50% chance of a girl and 50% chance of a sex determination in hen. In hen, the male does not decide the sex of the progeny, but the female decides the sex of the progeny. Female is dominant. So Z, 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 W. So here the males would be AA plus ZZ and the females would be AA plus ZW. And the gamete sperm will be of only one type, that is A plus Z. But from females, we can have two types of egg, that is A plus Z and plus W. So here the heterogametic parent is female, which will decide the sex of the progeny. Now, sex determination in honeybee. Honeybee, you know beta, in honeybee we have three kind of varieties. Worker bee, which are diploid sterile females which cannot undergo reproduction. We have queen, which is a fertile female which is diploid, and we have drones, which are fertile males but they are haploid. Okay, so in case of honeybee, the females are diploid having 32 chromosomes and the males are haploid having 60 chromosomes. Okay, so during gamete formation, what will happen here? Meiosis will happen. And we know meiosis is a reductional division, right? Reductional division. So after the reductional division, the female gametes formed here will be having how many number of chromosomes? 16. Okay, 16. So the female gametes are haploid and have 16 chromosomes. But in case of males, gamete formation is not by meiosis, because meiosis cannot occur in a haploid cell. So here the gamete formation will be by mitosis itself. Gamete formation will be by mitosis, and it is an equational division. So the sperm would be having 16 chromosomes. Okay, so sperm and egg both would be having 16 chromosomes, but the egg are the product of meiotic division and the sperm are the product of mitotic division. Now, whenever this female gamete remains unfertilized, when this female gamete remains unfertilized and directly forms, without fertilization, the female gamete directly forms an individual. If it directly forms an offspring, okay, if the female gamete without fertilization directly forms the offspring, then this offspring will always be a male, will always be a drone. And if the female gamete fuses with the male gamete, that means whenever fertilization will occur, the progeny will always be a female. Okay, so whenever a female gamete fuses with male gamete, the progeny formed will always be a diploid female. But whenever the female gamete without fertilization directly forms the offspring, then it will always be a drone. And that is why drones are always haploid, because they are the product of parthenogenesis. They are not formed by fertilization. And hence, the sex determination in honeybee is also called as haplodiploidy or archenotochy or parthenogenesis. So who is the product of parthenogenesis? The drones, the males, which are always haploid. Okay, beta. The drones are always the product of parthenogenesis formed by unfertilized egg, and the females are always the product of fertilization formed by fusion of gametes. So this is the topics are finished, and I hope this will help you in your term 1 exam, and all the best for your term 1 exam. Thank you all. Thank you.